longitudinal follow-ups. So we collected that data from their files, and as ma'am had already discussed, their mean age at onset was around 26, age at diagnosis was around 33, and mainly they presented with foot weakness, difficulty in dorsiflexing or lifting their foot. There was difficulty of proximal muscles, that is difficulty in getting up from the floor, difficulty in climbing steps in around 32. Distal upper limb weakness causing hand grip weakness was present in 8. Some of them had an asymmetrical onset, that is one side was earlier affected than the other. And about these mutations also ma'am has already told. So homozygous means that there are the same variant is present in both the alleles of the gene. Heterozygous means compound heterozygous means two different variants in the two alleles of the gene. So in Indian population, so it was more of compound heterozygous mutations compared to the homozygous ones. And among the homozygous one, it was the Roma gypsy founded mutation, which was more common. And also, but among the heterozygous ones, it was in the Indian founder mutation, with one allele having this mutation present in at least 131 of them. So with a follow-up of around 3 years, the loss of independent ambulation was seen in 29 of them, assistive devices in 20, wheelchair-bound state in 14, and bed-bound state in 4. And this occurred nearly 12 plus minus 4 years after the disease onset. So we find that, so in that study also the data on the various functional scales like muscular dystrophy functional rating scale, the IBM functional rating scale were collected. So this also showed progressive decline. But none of these patients on follow-up had any cardiac or respiratory symptoms. And also some correlation was attempted between which domain of the enzyme was affected, the kinase or the epimerase. So it was found that the kinase domain had a more severe involvement, upper limb involvement, as well as age of onset, initial presentation, age of first use of assistive device, all were more common in the kinase domain. Whereas the Roma gypsy mutation had a more severe phenotype, the Indian founder mutation had a lesser or a milder progression compared to the Roma gypsy one. So this was another patient registry of the GNE myopathy patients from Japan. So this was a part of the Treat NMD Alliance. So 121 patients were included, and also majority were female, 66 females. And also on follow-up, it was found that 52 had become non-ambulant, whereas 24 remained ambulant without any assistance. The respiratory function also showed some decline in around 53% of the patients, whereas the cardiac function test available in 35 did not show any decline. So building such a nationwide registry is very important in disseminating information to the patient, for patient recruitment, for resolving any data deviation, because if it is a single institute study, there might be patients who have a more serious or a more advanced disease who may present to the specialized centers. So if we have nationwide registries, we can include more patients. So, and also any data deviation may be avoided. And these were the common symptoms of these patients. All presented, majority presented with walking difficulties, difficulty in lifting toes. Some had difficulty in climbing stairs. And on follow-up, it was found that the assistive device use occurred around 12 years after the disease onset, wheelchair use around 15 years after disease onset, and the loss of ambulation occurred around 21 years after the disease onset. This was another model in which they tried to develop a disease model progression in GNE myopathy, so using longitudinal quantitative muscle strength data. So understanding this is very important for clinical trials, because otherwise we may not be able to provide a definite answer about the effectiveness of a therapy. So in this study, they followed up 38 patients for about mean of 14 months, ranging up to 35 months. That is, total cumulative follow-up was around 44 years. So, and they found that, as you can see in this red line, which is shown by the pointer. So here, this is the ankle dorsiflexion. That means the muscles involved in lifting the foot, which are very severely affected very early in the disease. And followed by when we see the knee flexion as well as the elbow flexion, which are marked by these lines respectively, they show the next decrease. But these decreases happened around 20 years and 30 years into the disease onset respectively. Whereas when we find the knee extension, which is marked by this gray line, it remained almost stable or showed very slow decline even into 40 years after the disease onset. So this is another international multi-center study. It was also a non-interventional observational study by Professor Hans Lockmuller and group. So this was again built up for building a larger group of the GNE myopathy community. So they had an online study as well as an in-clinic assessment. So this was published in 2021, the result of this in-clinic assessment where the patients underwent 6 monthly assessments for a period of 3 years. So what all assessment was done? They did the muscle strength, handheld dynamometer, the grip assessment, 6-minute walk test, assessing the various activities of daily living using scales like GNE myopathy functional activity scale. Among 101 patients enrolled, 87 could complete the follow-up up to at least certain point of time. So we can find that the mean age was 39, and among these patients, so 46 could complete walking for more than 200 meters. The age at first symptom onset was 26, the most common symptom being tripping. And also we can find that on serial assessments of the handheld dynamometry for assessing the hand grip and other muscle strength, there was a decline by around 3.8 score at the end of 36 months of follow-up. Similarly, the lower extremity also showed a functional decline. And among the muscles which showed maximum decrement, the statistical significance was seen for elbow flexors, elbow extensors, hip flexor, hip extensors, and knee flexors at 36 months of follow-up. This is another 2022 study which was again from the Japanese group. So wherein we can find that the age at closing ambulation occurred around 35 plus minus 12.4. And these are the various charts which are showing the various assessments, the manual muscle testing, the 6-minute walk test, handheld dynamometry, grip power, pinch power, the respiratory assessment using the forced vital capacity, and they also assess the creatine kinase and various other functional scales which showed a slow decline up into their 5 years of follow-up. So now coming on to what we are doing in the current natural history study. Why do we need this study? Because we know that there are certain genotypes which are common, and these genotypes will be different from what is present in the Western population or from the Japanese population. Currently, there is no treatment, though there are hopeful upcoming therapies. And for testing any possible therapies which may come up, we should have reliable quantitative models of the disease progression. This is especially important because GNE myopathy is a very slowly progressive disease. So, and also as I told you, there are no data from India on serial longitudinal assessment. So there can be population-dependent features which affect the progression of the disease. The genotype and the phenotype of the Indian population may be different from those in the other parts of the world. So our objective was to obtain data regarding the disease progression and also about the ability to perform activities of daily living and to derive functional ranking, to evaluate the muscle strength of selected muscle groups through physical examination over a 2-year period, to analyze the possible genotype-phenotype correlation and to study the biomarkers. So currently this is a single center prospective study being done at NIMHANS. So the evaluation of these patients is being done at 6-month intervals for 2 years. 50 patients with genetic confirmation who are ambulant are proposed to be included in this study, and the funding was provided by World Without GNE Myopathy, and ethics approval was obtained. And after recruitment of the project staff, we have an occupational therapist who is dedicated for this project. The study was finally started in November 18, 2024. So what will we do as a part of this study? So we have a questionnaire. So I'm just showing a part of the questionnaire. We ask the symptoms regarding various, we ask questions regarding the various symptoms. Foot drop, proximal muscle weakness, hand grip weakness, difficulties in running, jumping. So if yes, what is the duration of each of these? So we take a detailed questionnaire. We do a detailed clinical examination. To assess how are the activities of daily living affected, we have various scales. This is one of the GNE myopathy functional activity scale, which we have little adapted to the Indian population. So as you can see, there are various parameters like turning in bed, sitting up, walking, climbing stairs, various hand function assessments like mixing food, carrying objects. So this is scale graded on a scale of 4. So if the patient doesn't have any limitation at all, then the maximum score is 4. But if he is unable to do it at all or requiring maximum assistance of a person, it is 0. So the higher the score, it is better, which means it is milder. So it is a 25-point scale. So each being graded from 0 to 4. So likewise, we do the IBM-FRS, which is again another 10-point scale, which assesses the various activities, bulbar, hand functions, and like muscle assessments. And this is the modified Rankin scale. Again, all these are scales for assessing the disability. Then we do a detailed clinical examination using the MRC muscle power scale. So we test the muscles, how good is their strength. So this is again scored from 0 to 5, 5 being the normal, 0 means they are totally weak. So we do the assessment of the hand function test using dynamometer. This is a handheld dynamometer. So in which the patient exerts the pressure and also with the force of exertion that is recorded in pounds or in kilograms. And here this is again a pinch gauge in which we can again assess what is the strength of their pinch. So these are again various tests of hand function. The next is how is their walking, which is assessed using a gait assessment. So this is something which is known as gait ride, which we have in the physiotherapy department here. This is an electronic automated walk pathway with special softwares. So the patient, so there is an electronic mat which is around 4 meters wide and which is around 0.9 meters, 40 meters long, 4 meters long and 0.9 meters wide. So the patient is asked to walk on it, and this, with the help of this software, we'll be able to know various temporal as well as spatial gait parameters, like what is the step length, how many steps are taken, and also what is the length between each step, the length between each time one foot leaves the ground to the next time it touches the ground. So how much is the pressure exerted by the heel? So there are various electronic sensors which are present in this mat. So these electronic sensors, around 18,000 are there. So these will be sensing and we can know the different gait parameters. Another thing is the 6-minute walk test, in which in a 30-meter long veranda or a balcony, we are making the patient walk. So in 6 minutes, how much distance the patient is able to walk. Along with, it is a measure of the aerobic endurance. So we also assess the pulse rate, their breathing, as well as the distance walked during this. So this is one of the studies on the spatiotemporal gait characteristics using the gait ride, which we have published before. So there are particular characteristics for the GNE myopathy patients and with difference in the pressure which is exerted at the heel. And this is the MRI muscle. So all these other assessments which I mentioned, the muscle power charting, the dynamometer, as well as the gait assessments, all these will be done at every 6-month interval. This is a representative image of the muscle MRI. So as you can see that as the time progresses, you can see this is the normal appearance of the muscle, and these muscles which appear as white are the ones which have undergone fatty infiltration. So as the disease duration progresses, the muscles undergo fatty fibrous tissue replacement. So we do a muscle MRI yearly to know the degree of fatty infiltration. These are some of the images which are taken from one of our patients. So currently this is the images from the thigh. You can see that the muscles in the posterior part of the thigh are showing some fatty degeneration, as you can see here, as well as the muscles in the anterior part of the leg are also showing significant fatty changes. Of course, as I mentioned, TICS, our collaborating institute, does some blood and urine samples for biomarker analysis. As you know, it's a hypoxylating disorder. So we're trying to find some biomarkers. So currently we have enrolled 25 patients in this past 3.5 months, and we expect to complete the remaining recruitment within May. So it is through the support of the patients who are willing to come to us. So at any phone call, they're willing to come from even such long distances, and because they wish to find a cure for this disease, that we are able to carry forward this research. This is the collaborating research team, myself, my guide and mentor, Professor Nalini, Dr. Saraswati. This is Dr. Jitendra Saini, who is the neuroradiologist who will be doing the assessments. And of course, the main credit goes to our occupational therapist, Mr. Sugantha, who ensures that all the patients who come here have a hassle-free experience. He makes it smooth for them. Professor Rajesh Iyer and Dr. Sanjay Lamba from TICS. Of course, without the support of Professor Bhattacharya, Sudha ma'am and Dr. Rakesh Misra, we won't have been able to carry it forward. So, but the main thanks, as I mentioned, to the patient community here who are willing to come at all the 6-month intervals. So we hope that we'll be able to complete the recruitment as we plan, and we hope to have your continued support for ensuring a smooth running of this project so that we get some good data on the natural history. And also we hope that the sooner the drug comes, we'll be also able to help you in getting those. Thank you.
Thank you so much, Dr. Seena. That was very efficiently, also on time, from a time perspective. Thank you so much. So we have one question from Dr. Mishra, and if there are any other questions, please do put it in the chat box or raise your hand. So Dr. Mishra is asking, what kind of timelines we have to reach a point where a trial can be designed based on the natural history study? Are we getting some ideas from the cases from other countries as some features can be similar? Can we use AI mathematical modeling to accelerate the study?
So regarding what kind of timeline we have to reach a point where a trial can be designed. So I think, see, with the possible upcoming therapies, I'm not sure of the timeline, but at least we need a data of nearly 2 to 3 years if we have to really comment about it. And as I mentioned before, for a natural history study, ideally the duration is longer. But as based upon the disease model progression, which I showed in one of the studies by Dr. Quintana et al., so maybe we'll be able to develop a disease model progression, but I think 2 years should be the minimum we should be getting. From the other countries, of course, there is a difference in the genotype because our Indian founder mutations and in India, as we found, it is compound heterozygous mutations are more common, whereas in some of the countries, they have homozygous mutations which are common. The genotypes are different. So that way, I think we have to have our own Indian data or maybe some extent of comparison may be possible. Mathematical modeling and things, I think Dr. Rajesh sir and others will be also helping us with that to know how we can accelerate this. So I don't know whether anybody who has participated in the study is already here. So of course, Dr. I think somebody Mansi Khare is asking. So I think we have called almost all the patients. I am very sorry that we haven't received a call, maybe for some reason your phone was not reachable or why, I don't know. We have actually made a cohort of the NIMHANS patients as well as some of the patients from WWGM who are also a part of the existing NIMHANS cohort, as well as some who are not a part of the existing cohort. We had actually called up from all the groups, but I don't know how your name might have been missed. But of course, we are continuing the calls. So if somebody is not, all the patients, some of the remaining ones, they are telling they require time to think when they will be able to come, April, May. So we continue to call all the patients whom we have missed out. So we'll ensure that we also receive a call soon. Okay. So somebody who has received enrolled in AIMS, fine. So whichever is more convenient to you, that is also fine. Okay. So but
Thank
naturally,
you. I'm on the call, so on Mansi here. So I'm looking forward to coming and have no problems traveling to
sure,
Bangalore.
sure. We'll definitely call you and we will make sure you get a call soon and you're enrolled in the study. Thank you.
Thank you.
Yeah, Shilpi, I'll like to answer this question, Rakesh's.
Yes.
So, Rakesh, I think, we will need minimum 3 sets of data, that is 1.5 years of data before model can be start developing. And then probably initial model Sanjay can develop and then it can be subsequently modified as more and more data comes in. And then once we start getting the model, then only we can compare with this Quentin et al.'s paper and see whether this thing matches with what they have. But I think 1.5 years is probably 3 rounds of data is probably needed before we can actually start seeing.
Because disease progression, if there is some consistency, we anyway have patients of different age group, so we can try to model that.
Yeah, so that sort of thing we can do. And best part for clinical trial would be that we have the same patient data in terms of progression.
Yeah.
We can use that instead of having a placebo control, actually.
Correct. Yeah. That's true.
So that would be the best part.
Yeah.
Okay, thanks.
Thank you so much, Dr. Seena, for such a comprehensive overview of your study as well as in general the natural history studies that have been conducted in the past. And I hope that also maybe with respect to, there is some confusion in the patient group between the AIMS one now and NIMHANS, so maybe hopefully that can be clarified later also. Dr. Vishnu can maybe clarify that at a later point. So now I would like to invite our next speaker for today, Dr. Rajesh Iyer, who is a senior scientist at TIGS and is working on a very cutting edge therapeutic technology, which is now sort of maybe has some very, very promising applications for rare diseases like GNE myopathy. So over to you, Dr. Iyer.
Can you hear me? Can you hear me?
you're a bit soft. I don't know, is it just me?
Yeah, low.
Yeah.
Better now.
Yeah. Now better?
Yeah.
Okay. Thank you, organizer, WWGM, Shilpi Bhattacharya, Alok Bhattacharya, and Sudha Bhattacharya for considering me to present the work. And thanks for the funding, because without WWGM support, the work would not be possible. I'm going to talk about the mRNA biotherapeutic platform for rare genetic disorders, especially with the focus of the end for GNE myopathy. We will start with something which is the COVID-19 pandemic, which got us the mRNA vaccines. These mRNA vaccines were developed within months of the COVID genome was sequenced, and it was quite efficacious in preventing the infection in many number of people. There was a phase 3 trial of more than 30 or 40,000 people, and it was quite efficacious. There was a quite fast turnaround time, as well as a very highly efficacious vaccine, which was not heard of before. Right. So basically, modes of therapy in the case of any kind of a rare genetic disease, or maybe any disease, it can be either DNA, which is more of a gene therapy approach. It can be RNA, which we are going to talk about, in mRNA-based therapies. In this also comes the antisense oligonucleotides, which regulate and control the mRNA. The third is protein, where the protein, which is the causative agent of the disease, where in certain kinds of cases, the protein is not functional, or certain kinds of cases, the protein is overactive, whatever it may be, where people start giving the proteins directly to the patients, or give them small molecules which can modulate these proteins. Right. So in our I'm not going to talk about gene therapy, but we specifically want to work on mRNA therapy, because this transition which happens from DNA to RNA to protein, it's actually, if you have to do a kind of a therapy for a rare genetic person, all these steps has to be done outside. Somebody has to make DNA, somebody has to make RNA, somebody has to make protein, and a lot of cost is invested in it. And ultimately, the rare disease drug becomes overtly pricey. We wanted to remove one step, and we wanted to focus on mRNA, because mRNA had a technology platform by which we knew that there were so many vaccines were being given, and indeed, it was safe and efficacious for many number of people. Right. So the first key innovation via the mRNA therapy was there from 1980s and 1990s. The first key innovation which led to Nobel Prize discovery was how to inject mRNA into a patient so that, without any kind of side effects. And that's what the Nobel Prize effect was there, where they found that if you modify the RNA, the entire of the RNA becomes safe for clinical application. The second key innovation is that once you make an mRNA, what is the next step? How would you deliver inside the patient's body? You need a vehicle, and the vehicle is lipid nanoparticles. These lipid nanoparticles encapsulate mRNA, sequester them, and when you put it inside the patient's body, it actually takes it to the right places so that the protein which is lacked by those patients can be produced. Both of these innovations actually led to the mRNA vaccine, where the mRNA vaccine encoded in LNPs actually carried the spike mRNA. The spike mRNA went inside, made protein, and actually triggered the entire of the immune system to provide the protective efficiency against the vaccine. Right. So I'm coming back to the mRNA vaccine point again, that it was quite efficacious, and coming back to it, we thought, why not we use an mRNA biotherapeutic platform, modulate the entire of the platform for rare genetic diseases. Why? Because there are 7,000 or more rare genetic diseases in India. And if you combine all the people of rare disease lived in one country, it would be more than world's third populous country. And there were so many diseases there. And if you put further point, there are around 1.2 million newborn affected by genetic disorder in India per year. And for most of them, there is no treatment available. And for few of them where the treatment is available, it's quite costly. So, case in point, let it be a Pompe disease, where you can see that it happens because of a mutation in a gene called as acid alpha glucosidase. And the only therapy available for it is an enzyme replacement therapy. The cost of this therapy is 20 to 40 lakhs per year for a 10 kg child. And thinking about this goes in crores and crores. And the maximum amount of money which Government of India provides is 50 lakh, which will not even cut for 2 years or 3 years of their treatment. We thought, why not we convert into an mRNA-based therapy, where the mRNA which is being there, which is lacked by these patients, mRNA can be made, which can code for these protein lacked by this patient. We encapsulate into a lipid nanoparticle. We actually check it in mice models, and efficiently, once we know that it is working in the mice, we take it to humans for phase 1 clinical trials. What is the promise of mRNA technology? The promise of mRNA technology lies in the sheer fact that it's a pipeline process. by a single process where you have mRNA synthesis, mRNA purification, encapsulation thing, if these 4 square boxes are ticked, all the other processes, all modes of therapy is open for rare genetic diseases. Maybe a protein production where you want to produce the protein lacked by the
Rajesh,
patient. Huh?
you
Yeah.
are showing presenter's view. So we
Oh,
are seeing the next slide also. If you close that, it's this it will become more larger,
is
easier.
it fine? Okay.
No, you have to go from the slide show. Their presenter view you have picked. So if you have to.
Okay. I'm sorry, I'll just see that.
Or don't
I
worry.
can help you. Go to display settings on the top and then just toggle them on the top.
On the top.
On your screen, there's something called display settings.
Okay, I cannot see display settings. Video
We
settings.
can see on the screen.
Okay.
Next to show taskbar
Next,
on
yeah.
the top.
Yeah. Next to that, display settings.
Okay, I cannot. Just let me just see.
Just
Okay.
next to, oh.
Now go to PowerPoint.
Am I? Is it okay?
You can just make it full screen.
Okay,
So
I just.
the presentation mode on the bottom of your screen.
It's not in
Okay.
presentation mode. It actually is in edit mode.
I think
Okay.
Coming back then. Okay, I think let's continue.
It's good.
Hide presenter view.
yeah,
Yeah, I did it.
That's it. Yeah, yeah.
Okay. Yeah. So
Okay.
sorry for the thing. So yeah. So why we wanted to make an what is it? Oh, sorry. Just wait. Yeah. So what we wanted is that in this, if you can, in the mRNA technology, the promise was, if in the square box, all these processes can be standardized, you can achieve different modes of therapy. Maybe it can be a protein replacement therapy where the patient can be given the protein, where the protein can be made inside the patient body, which can actually act as the therapy. The second, the same mRNA technology can be used for gene editing, so a single shot cure can be done. Or the same mRNA can be used for making an in vivo antibody treatment, because in many number of time it has been seen in cancers as well as many other rare genetic disorders. You can actually make antibodies and they can be used for therapeutic options. So all these processes can be done through a single square. And if you optimize this process, all the options opens for you. So entire of the thing, what happens is that the drug discovery cost plummets, and only with a single pipeline, you can make drugs not for one disease, you are making drugs for thousands of diseases. And when you combine all of them together, the cost of the drugs completely reduces, right? What is the other value proposition? All of the mRNA technology which we use is actually safe and clinically validated. So because billions of people have taken the dose of mRNA vaccines. And since it is a completely synthetic nature of manufacturing, means it does not involve cell lines, serum, and all those things. So the cost even further reduces. If you look at the point, for making an mRNA vaccine by Moderna in the US, it costs only $0.20 per dose. And if you put in Indian prices, it's way further lower. Right. Many people talk about like what is the difference between an mRNA therapy and a gene therapy, because it's quite a recent trend where people are really talking about gene therapies a lot. The gene therapies and mRNA therapy differentiate in one of the three things. The gene therapy is usually a viral-based therapy, while mRNA therapy is actually a non-viral-based therapies, where you actually encapsulate mRNA and a lipid, which is completely safe. Second, gene therapy, since you use virus, the body mounts an immune response against it. With LNPs, you actually have a less immune response, right? The good thing about gene therapy is that you can actually do it in only once and twice and maybe done with it. But it is very difficult for a disease like GNE myopathy, because where you're looking at very late stage progression of the disease, at that time, giving viral vectors is something which is challenging. In the mRNA therapy, it's a transient therapy which we can do weekly and bi-weekly as you can take a drug. The cost, I hope that Dr. Arkasubhra Ghosh at India can actually bring the AAV cost a lot. But right now, for Georgian ESMA, for SMA, it is around 14 crores. But what we think with an mRNA therapy, we can actually bring a dose to around 1,000 rupees or maybe even lower, right? So this is the kind of an interface where we are trying to work with respect to mRNA therapy. Right. So what is the global status of mRNA therapy? The global status is that the world is catching on. There are lots of clinical trials which is being done by Moderna and Vertex and Translate Bio, which are going on for mRNA therapies, especially for propionic acidemia, methylmalonic acidemia, cystic fibrosis, and etc. There is one great therapy which is actually now in phase 3 clinical trials where actually do a in vivo gene editing through mRNAs. We are also attempting this. Probably I will slightly twitch up the data which we are trying to do for GNE myopathy as well. There are lots of companies. There are some Indian companies like Biological E as well as Genova Biopharmaceuticals which are being involved in things. And there are lots of other people like Pfizer and Sanofi involved in the making mRNA drug development. Right. Where are we? What are we doing? So we are in the lab. We actually work on 4 diseases. Actually, they work on Pompe, Fabry, Gaucher, and monoclonal antibodies, where we are actually making the mRNA in which the protein which is lacked by these patients would be made through mRNA, which would actually go to the liver, produce the protein, and go into systemic circulation, just like an enzyme replacement therapy, so that the cure can be caused. The other which we are working focused on is through WWGM sponsored project and also an ICMR project where we are trying to make the protein which in the GNE myopathy patient, the GNE protein which is being lacked by it, we actually make an mRNA for it. We actually give it to the patients. Actually, we want to give it as a therapy in the muscle so that the mRNA can translate inside the muscle to make the protein, and the kind of the absence of the protein can be complemented and the disease can be halted. We actually also work on Cas9 mediated stuff where we are looking at in vivo gene editing through mRNA. This is more of an where it's a single shot treatment, while the other treatment was far more recurrent, while this is a single shot treatment. Right. So usually as a test of any kind of therapy, we have to take to an entire of the preclinical journey. And this is how we do it. We actually make an mRNA, we purify the mRNA, we encapsulate lipid nanoparticles, and we actually check. So let's check that what we are making is true or not. Right. So in this, what we have done is that we have taken an mRNA which codes for a light emitting protein. So if you see that when we make this mRNA, encapsulate into a packaging vehicle and give it to the mice, you can see that the liver consistently picks up that signal, and the light emitting protein actually produces the light which can be captured, showing that in principle, whatever we're working on can work in mice and probably can work in human itself. So with the focus that once we had the technology in our hand, we actually focused on lysosomal storage disorders, because we knew that when lysosomal storage disorder, enzyme replacement therapy works, means the protein which is lacked by these patients can be given intravenously. So we thought, why not we give this protein, just like as I told you before, through the mRNA. And then we tried this thing in mice. First of all, we tried it in cell lines, where we actually make this mRNA, and we found that the enzyme, the protein which we can be made in cell lines very nicely, as well as you can see that the enzyme is active. That whatever the protein which is being produced by the mRNA, it's not functionally defective, it is active and can actually go ahead. And if we inject inside the mice, actually in the liver of the mice, we can produce the protein, showing that a proof of concept which we have achieved right now, that if we make an therapeutic protein, if we make a therapeutic mRNA, encapsulate lipid nanoparticle, it can produce, it can, and give it to mice, it can allow the production of protein inside the mice liver. This is our preclinical proof of concept. And as I told you that the mRNA therapy is extremely parallel therapy. Actually, it is a pipeline kind of thing. If you optimize it for one disease, for other 10 diseases, all the avenues opens up. So we took all the application and design processes which were there for Pompe, and we actually took it to Fabry. And when we did it to Fabry, we again found that we can achieve the proof of concept with respect to the Fabry disease as well. So right now, with both of the Pompe and Fabry disease, we have preclinical success at TICs. Now, coming back to the GNE things where we are trying to focus, we wanted to do the same thing for GNE myopathy, but the GNE myopathy is actually a different kind of a disease to work with, because GNE myopathy affects muscle, where in the case of all the lysosomal storage disorders, we are looking at liver to produce the protein and reach to a different part of things. But in the case of GNE myopathy, we have to actually focus on a specific muscle. I don't need to go about all the kind of the disease and stuff, but the key thing which we wanted to highlight is that the disease happens due to mutation in the GNE gene, and we want to replace this gene by actually mRNA, the mRNA which are being encoding for the GNE protein, mix it with lipids, and actually use this lipid nanoparticle formulation inside the patient muscles so that the GNE protein can be produced. So these are the objectives of the studies which have been trying to do that. It's for this we have been talking to Alok a lot and the ICMR consortium also, where we are actually making GNA encoding mRNA. We are actually checking in the cell lines, and we are making muscle targeting nanoparticles and checking in wild type and mice models. So just to go through some parts of data which we have generated for GNA, we made the GNA mRNA, and we actually take this GNA mRNA and gave it to cells and see whether the protein can be formed or not. And if you can see it here in two different cell lines where this control which is not being transfected, in the case of this is the GNE protein which is being lacked by the patients, and we can actually make this protein inside the cell lines. So this is something which once we have this RNA which can produce the protein, the next step is that whether we can take this mRNA inside the muscles so that the protein can be made in the muscles of the patient. So for this, we took a very specific kind of approach with mice because it's very difficult how to give specifically to muscles, right? How you actually can take any patient or and inject them with the drug, and how would we specifically go to their muscles. So for this, we did a preclinical study where actually made muscle targeting nanoparticle formulations. And what we did is again, we use a light emitting protein, and we actually gave it to mice. And as you can find that when we inject into mice, it is specifically there in the hind muscles only at 2 doses. This actually gives us a proof of concept that we can make a nanoparticle formulation in which we can put a GNA encoding mRNA, encapsulate them, and gives us a hope that if we give it to the patient's muscle, the protein would be produced. And hence, when the GNA protein is being produced, the deficiency of the GNA would not be there. And hence, the disease would be halted or mitigated. So this is the stage we are in. Right now, we are conducting preclinical therapies for the GNE, where we are actually injecting the mice with GNE protein and seeing how much of the GNE protein is being produced in the mice. And this is something which is an ongoing process, and hopefully we will have a therapy very soon for the GNE, at least a preclinical proof of concept, which can be taken up by the drug regulators for the next stage. Apart from that, we also spoke about single modal therapy, where we actually use an editing enzymes. So for this, we are actually using the gypsy mutation, the Rajasthani gypsy mutation, and we are seeing whether can we correct this mutation inside the patient. And this is something is an ongoing process again, where we have taken cell lines, and this is actually a control cell line in which there is nothing which is there with respect to the, we go and edit the GNE gene. In the control where there is no editing, but here when we use a lipid nanoparticle with the editing enzymes, we can actually edit 70% or 80% of the cells. And this is the place where we are trying to go, where we can we take patients with harboring mutation like Rajasthani mutation or valine 727 mutations, can we give these lipid nanoparticles so that the protein can go inside and actually correct the mutation so that the disease therapy can be obtained in single outcome, quite akin like gene therapy. We are doing we have done it for cell lines, and we actually have done it in mice also, and we are seeing positive results, and we have to see how it goes by in the coming times. So this is something which I just wanted to show at what we are trying to do, and we are right now in the lipid nanoparticle formulation, the experiment wild type mice, and this is something which is progress, and we are thinking that we will reach the milestone very faster. Similarly, for Fabry and Pompe, we have reached a place where we have done the POC and we are looking at preclinical efficiency. We will reach for GNE myopathy soon, and we also start to do a preclinical efficiency study, and actually with the aim to show that the entire drug is safe so that it can be taken for the next stage of clinical validation with the help of WWGM and ICM. Right. Many people spoke about it. Dr. Rakesh spoke about it. Dr. Alok spoke about it. Dr. Seena also spoke about it. Nalini spoke about it. That these are all, we are all part of a Center for Advanced Research for Neuromuscular Genetic Disorders. And here we are using, ICMR has told us that if we can actually make a therapy for rare genetic disease, they would actually take it for clinical trials. There are 4 main objectives here, which are mRNA-based therapeutic gene editing candidate, making mRNA-based therapeutic candidate diagnostic assays, doing cellular and animal models, and doing clinical modeling. clinical modeling, Dr. Seena as well as Dr. Nalini spoke about, and that is part of the studies. And cellular and animal model is also very important, because when we make a therapy, how would we know that it's working? We don't want to directly go to the patients. We want to first of all go and check that it's cell line it's working, it is working in animal, it is working in multiple sources, so that when we present this therapy to a drug controller, they know that we have tested all the avenues of science, and then we are producing a safe drug which can be taken for clinical validation. So impact, I don't need to say the impact which we'll be having for the therapy. GNE myopathy, it has no kind of thing. And if you actually can make a drug for it, it is phenomenal. And the good part of mRNA therapy is that if you make the drug for one, you're not looking at one disease, you're actually looking for multiple diseases. And these are my collaborators, and I thank all of you for this opportunity. Thank you.
Thank you so much, Dr. Iyer. And I have to say, I'm so impressed with all the speakers sticking to time. Like, oh, I mean, yeah, it's very, very impressive. Your dot on time, Dr.
Thank
Iyer.
you. Thank you.
So your talk has led to a lot of excitement in the patient community. There are a number of questions about the treatment. I know that I think most of these are too, like, right now it's a bit preliminary, but do you want to take up some of these questions?
Yeah,
And if,
please.
and if anyone else has questions, please do ask. So the number of questions on cost of treatment and will it help rebuild lost muscle, and whether it will, the other question was on whether it's a one-time treatment or not.
Okay, so right now the therapy which we are developing, it's we are actually looking for both the kinds of therapies, because we are looking at a kind of weekly and bi-weekly dosing regimen where we can produce the protein and give to the patients, every monthly or for every 15 days. The other mode of therapy which we are looking at, a single shot kind of therapy, which is in the progression, which would have a more developmental timeline than the recurrent therapy which we are looking at. We are positive that when we do this therapy, the disease symptoms would halt. We do not know whether it would go back to a place where the muscles would regenerate. But right now, our aim is whether we can halt the disease. And in the case of GNE myopathy, we are given a big therapeutic window. So if it starts around 2025 and it goes till 30, and then the results are presented. If we can actually develop a therapy for patient and administer them at that place, we are hopeful that they would be completely normal and no muscle phenotype would be appearing in them. And that is the place we are looking at therapy. At certain point of time, when we have achieved this objective, yes, we would be looking at a certain kind of places where we can regenerate it back. That is something which mRNA allows us, because then we were not looking at just GNE as a protein. We are looking at some other proteins also, which can allow muscle regeneration. So that is something which we are not thinking about. Right now, it's recurrent therapy to halt the disease symptoms.
Side effects, there's a question on side effects.
Yes. So every disease would be every drug would have a side effects. It's not something which is very which we are very cognizant about, and that is what we keep on trying our drug in cell lines. We actually check in mice, and not even restrict to mice. We go to rats, we go to rabbits. We actually also take induced pluripotent stem lines where we actually use cell modeling, human cell modeling, to show that it is completely devoid of any kind of toxicity. The other part which which also gives us confidence is that the kind of formulation which we use is quite similar to modern mRNA vaccines, and those vaccines have been administered to billions of people, and we have seen very minimal side effects. So in that part of thing, we are quite we are quite confident that whatever therapy which we will be producing at the end would be completely vetted for toxicity, and the toxicity landscape is quite small right now for us.
So last question, Dr. Iyer, from Devinder. She's asking whether the muscle targeting LNPs, whether they're targeting all muscle types or specifically skeletal muscles.
We are looking again at skeletal muscles right now. Smooth muscles, it would be a next part of the things, because we are thinking that if we can administer the therapy locally at the distal muscles which are most affected, maybe we can actually rescue the symptoms of the disease. But later, at certain point of time, we are actually looking at some place by which we can actually use these particles to deliver to all the muscles. And when we do that, we are not just looking at GNE myopathy, we are looking at multiple number of neuromuscular genetic disorders, as Dr. Narayana pointed out, limb muscular genetic dystrophy, dysphalanopathies, all those things come into the picture. But right now for GNE myopathy, it's focused on the distal skeletal muscles.
You're muted, Dr. Mishra.
There was a question about cost. Can you comment?
Okay, so the cost of the therapy is, that's why I said the cost of the mRNA therapy for the Moderna mRNA vaccine was around, making it around, costed around $0.2 per dose. I think if we have to, we use that as a reference and we extrapolate it, we are looking at 500 to 1,000 rupees per dose. And this is for recurrent therapies. But if we are looking at something which is for one-shot therapies, if we are looking for one-shot therapies where we are trying to do with CRISPR-Cas9s and everything, within like 5,000 or 6,000 rupees, we can probably have an outcome for a patient, and that would be a single shot option. So we have to be really, we at TICS, we are very much committed to make an affordable care. We don't want to make any therapy which is beyond the scope of the common people in India. So that is something which we keep on focusing. And since we are from a not-for-profit institute, all the other avenues of research and profit comes completely out of it. So the drugs cost even further reduces. So we want to keep it very much affordable, and that would be Dr. Rakesh would make sure that that happens.
But your cost will not be that of vaccine.
Yeah, there's
Because your
scale is
dose
different.
will be much higher than what you give in vaccine.
Yes, I agreed. But what we are looking at labor costs, which are being there in the West labor costs. So in India, when we are looking at these manufacturing, this cost would dwindle down, and then that would take care of the extra doses which we are trying to give in the case of mRNA therapeutics.
No, also
So that
we can make it more translatable. I mean, in the sense that
yeah.
each of us, those things
Yeah.
can come. Anyway.
Yeah.
Okay, so there are a few other questions, Dr. Iyer, but could you maybe just respond to them in the chat box itself, because a lot of patients have questions, but we are a bit short of time, so I would like to move on to our next talk. So if I could just request you to respond to the questions in the chat box, that would be...
I would do so. I would do so. Thank you for giving me the opportunity. Thanks a lot.
Thank you so much for this really exciting talk. And I think the patients are so excited and they're looking forward to, I think,
they are the source of motivation for us, because when the things don't work, we just think about them. And that is something which is integral to us. Thank you very much.
Thank you so much. So with that, I would like to invite our next speaker, Ms. Divya Rao, who is doing her PhD in the lab of Dr. Munia Ganguli, who is a scientist at CSIR Institute of Genomics and Integrative Biology. Dr. Ganguli could not be here, but she's also doing very critical work on non-viral vector gene therapy, which actually Ms. Divya Rao is doing. So she's going to present her work. So we look forward to hearing the progress that has been made in non-viral vector-based gene therapies. Over to you, Ms. Rao.
So, I am audible?
Yes, you're very clear.
Okay.
If you can put the present, yeah, perfect.
Okay. Just hide this. It's fine. So I am really honored to share some of our lab work, non-viral vector-based gene delivery for GNE myopathy on this platform. And I will briefly introduce gene therapy, and then I'll go why did we choose non-viral delivery vector, and then I'll come to the work that we have done. It's not moving.
It's not moving.
Yeah, it's not moving. Wait.
Do you want, yeah, start it again, maybe.
No, it's not moving.
Do you want to share your slides with me?
I'll just try once more, or maybe I'll just leave and then rejoin.
Yeah, maybe you can do that.
You share the slide, don't share the screen, then it will work.
Yeah, she's left the meeting though, Dr. Mishra.
Okay, maybe she'll come back.
I'll just share the slides.
Yeah. Don't share the screen. Don't share your screen. Just
It's coming like the host isn't allowing to
Oh,
share slides.
sure. Baba, will you make her co-host? I can't make you co-host.
Now I can take.
See if you can move your slides right now.
I'll just try once. Yeah.
Yeah.
So I'll be just briefly about gene therapy. So basically, we do gene delivery in order to achieve gene therapy. And in gene therapy, these three different terms comes. For gene augmentation, it's basically when the gene is mutated, we and it's hypofunctional, then we deliver a functional copy of the gene so that the cell can function normally. And that's what we are doing in the case of GNE myopathy. In the case of gene silencing, sometimes the mutation leads to a formation of a hyperfunctional, can lead to the formation of some hyperfunctional gene, which produces the protein abruptly, which can, so for this, we need to deliver a blocking gene that can block the expression of this mutated gene so that the cell can behave normally. In the case of gene editing, nowadays people have even try using CRISPR-Cas9 in order to correct the mutation for in order to cure different kind of genetic disorders. Now, after after a brief introduction of gene therapy, we are just wanted to know how does one do gene therapy. So basically, gene by itself can't go into the target cell. So we need some kind of delivery vector to package them. And gene therapy can be done by ex vivo or by in vivo condition. In ex vivo, the mutated cells are taken from the patients and then they are being corrected. And then these modified cells are again reinjected into the patients. In case of in vivo conditions, the gene of interest is packaged to the delivery vector, which will then go to the patients. Now, when we are talking about gene therapy, there are three different key components for in order to carry out gene therapy. First one is delivery vector and the cargo and the targeting moieties. So delivery vectors is the area where our lab works on, and it's broadly classified into two different categories: viral vectors and non-viral vectors. And the cargo can be DNA, mRNA, siRNA, and the targeting moieties, as we are working for muscle delivery. So in that case, it can be antibodies or muscle targeting peptide that has specificity towards muscle receptors. So I'll just briefly introduce viral vectors. As over the years, researchers have explored these viral vectors. As you can see here, there are a lot many viral vectors that are in clinical trial. And even there are some AVs that have been explored for GNE gene delivery and that are in preclinical studies. But there are some limitations associated with AAV-based vectors, like the payload capacity is limited to 4 to 5 kb, and immunogenic response associated with it, which makes its dosing very critical. With all this, non-viral vectors can be alternative approach. We can use it, and we have summarized some of the non-viral vectors that have been used for different kind of muscle dystrophies like DMD and SMA. And we have broadly classified it into peptide-based non-viral vectors, lipid-based non-viral vectors, polymer-based non-viral vectors, and inorganic-based non-viral vectors. And these vectors even have been modified with muscle-targeting peptides in order to deliver gene or any cargo specifically to the muscles. And these all things give us an idea that even these non-viral vectors can be used for GNE myopathy. And there are this group which have worked on GNE gene delivery using lipid-based vector, but they have not follow up their studies. There are some challenges that comes in play when we talk about delivery vector. So if in case of muscles, there are two different routes that has to be by which we can deliver these nanoparticles. It is systemic injection or it can be intramuscular. For systemic injection, the main challenges I'll be discussing in my future direction slides, but basically from the blood vessels, it has to transcytose out, and then it has to diffuse through the extracellular matrix in order to reach to the target organ. And then it has to go to the nucleus. So keeping all these steps in the mind, the designing of a nanoparticle become critical, and where these shape, size, and even the targeting moieties play an important role. And keeping all these challenges in our mind, we have proposed two different strategies: peptide-based non-viral vector and the polymer-based non-viral vectors. I am spending one slide on these peptide-based non-viral vectors where we are working, but our main focus will be on the other strategy. So basically what we are doing is we are using a cell-penetrating peptide, which is positively charged, and we have a patent in our lab for this particular cell-penetrating peptide, and this is well known for gene delivery for various organs like skin and lungs. And we are for using it to forming a nanocomplex with the GNE DNA. And then this nanocomplex will be coated with muscle-targeted peptide modified chondroitin sulfate. Why we are using chondroitin sulfate? Because it has some properties like it can improve the biocompatibility of the nanocomplex, it can decreases the charge, it decreases the immunogenicity. And the muscle-targeting peptides that we are using are already known in the literatures and shows better gene specificity towards the muscle. And we are getting some promising results with the reporter plasmid, which we are going to validate further with the functional plasmid and also in vivo. Coming to our second delivery vector, where we are using PBA-based polymers, and we are forming the nanoparticles with the GNE gene. And right now we are giving intramuscular delivery, but in future plan, we are planning to make a systemic stable PBA polymer so that we can give systemic injections. So I'll just briefly go through why did we choose PBA polymer. So PBA polymer were used for gene delivery in 2000, and now its structure has been used and its topology has been modified in various ways in order to use for various applications, including gene delivery and even vaccine purpose. So its basic structure consists of diacrylate and the amine group. So it's because of the ester bond present it's there in the structures, it become a biodegradable, which make it more biocompatible and reduces its cytotoxicity. And these tertiary and primary secondary amine makes it positively charged so that it can electrostatically interact with the DNA. So we have concluded all the properties of the PBA polymers, and one of the key property of the PBA polymer is that it consists of various monomers, and by altering one monomer, we can use it we can alter even any monomers for our particular application. I'll just go through that PBA is being used for gene delivery. So this Daniel G. Anderson group has initially made the library by reacting diacrylate and hydrophilic amine group. And from their library, these few monomers stood out and they showed better gene delivery efficiency out of the other monomers. And the same group have also shown that the addition of the end cap at the core structure of PBA polymer can boost its transfection efficiency. And they have used different kind of end cap. And now they have, like nowadays, this is the recent paper came on 2022. They have started incorporating hydrophobic amine in the code chain, in the main PBA chain, so that the interaction of the PBA with the membrane can be enhanced. Keeping all these things in our mind, we have started our work with a linear PBA, a linear well-known PBA, as this PBA is very less underexplored in the case of muscle disorders. And for this work, what we are doing is, first we are synthesizing PBA polymer and we are characterizing the nanoparticles using by checking its size, charge, and shape. And then we are going to screen it in vitro in muscle cell models, and also going to check the cytotoxicity. And after this, we are going to do the in vivo screening. Right now, intramuscular injection, but as I said, we are going to do in vivo injection, the systemic injection. So to start our work, we should have polymer and we should have the plasmid construct. So for this polymer synthesis, we have reacted the diacrylate and the amine group, and we have formed the PBA polymer, and we have confirmed its synthesis using NMR, and the polymer was successfully synthesized. For the preparation of the construct, we have inserted our GNE gene into the expression vector. So this slide is just to show that our final vector will be having the GNE gene fused along with the GFP gene. Now, as we have the plasmid as well as the protein, the polymer, we are going to check if our polymer is forming the nanocomplex with the DNA. For this, we have made the nanoparticle, and we have used increasing weight by weight ratio of polymer to DNA, and we have checked its complexation ability. And we can see that from 20:1 weight by weight ratio, the polymer started forming the complex. And we also wanted to check if our DNA, our polymer is able to release the DNA in response to the anionic challenge. And for this, we have used heparin, increased weight of heparin. And we could see even at the weight of 0.25, the polymer started releasing the DNA. And with this, we had also checked some biophysical properties of our nanoparticle by checking the size. So across different weight by weight ratio, we checked the size was around 250 nanometer, and the PDI was also less than 0.25, which shows that our the solution of the nanoparticle was homogeneous in nature. And also the charge was around 30 millivolt across different weight by weight ratios. From there, we chose 50:1. From our preliminary data as well as from literature, and we have checked its stability, and the nanoparticle was stable up to 24 hours. Then we wanted to check if our nanoparticle is able to deliver the gene of interest to the muscle cells, and for this we have used these C2C12 muscle cells, and we checked the gene delivery efficiency, and here we are checking the GFP signal. With the reporter plasmid, our polymer was able to give around 60% transfection, which dropped with the use of functional plasmid. It is around 35%, but it is comparable to the positive control. Now, with this, we also wanted to check if our GNE gene is expressing in the muscle cell or not. And for this, we have used primary antibody against the GNE protein, and we have performed ICC and Western blotting, and we could see signal coming from the GNE protein, which confirms that our gene was expressing in the muscle cells after treating it with the nanoparticles. Now, with all this, we know that our polymer is able to deliver GNE gene, but we wanted to check if these nanoparticles are compatible for the muscle cell. For this, we have performed MTT assays, and our nanoparticle was showing around more than 90% cell viability. After this, we wanted to check the gene delivery efficiency of these nanoparticles in vivo. And for this, we are giving our first dose at day 0 and the second dose at day 1. And then we are euthanizing mice at day 3 and isolating the tibialis muscle. And then we are doing Western blotting and IF to check the GNE gene expression. So this was the pilot study where we have used a 10 plus 10 total 20 microgram dose. We could see some signal, but still we have to play around with the doses. And this we will be doing in future. Coming to the summary slide. So the PBA is able to the PBA is synthesized, and we have confirmed it through the NMR. And the PBA is forming nanocomplex with the DNA, and the size is around 250 nanometer, and it is positively charged, and it is stable across 24 hours. And then the PBA and DNA, these nanoparticles are able to deliver a GNE gene to the muscle cell, and we have confirmed it by flow cytometry and by ICC and Western blotting. And our nanoparticle is compatible to the muscle cell. And also we could see some signal from the mice, but still we need to validate it now further with the proper group and with different doses. For the future direction, we also wanted to check the mechanism of cellular entry so that we can come up with some novel PBA design. And we have to finish our these in vivo studies. And as all these data are done in healthy mice cell or healthy mice, so we wanted to we develop our C2C12 we wanted to develop our GNE knockout in C2C12 muscle cell using lenti-CRISPR V2 vector. Now our ultimate goal is if we can able to make a systemic stable PBA polymer, and for this there are key challenges that it has to face, like the rapid clearance by the immune system, the serum instability, and the non-specific biodistribution. Keeping the first two challenges in our mind, in our lab, we have developed a systemic stable PBA polymer for gene delivery to the brain. And for this, we have used hydrophobic amine and the PEG so that we can make a systemic stable PBA. And the same strategy we are going to use to develop our polymer. And the other thing is, as we know that if we give the systemic injection, most of the nanoparticle goes to the liver. So we are planning, and there is this paper which came in 2021 where they have substituted the diacrylate with novel diacrylate that was bisphenol ethoxylate diacrylate, which shows liver detargeting efficiency. So we are thinking if we can incorporate this particular diacrylate into our design. And the third thing is we are screening different kind of hydrophobic amine so that it shows better specificity towards the muscle cells so that it can go to the muscle and not to the other organ like spleen and kidney. So the overall future direction is if we can make a novel PBA design that is systemic stable and that shows some specificity toward the muscles. In the last, I would like to acknowledge my supervisor and Professor Bhattacharya, Alok Bhattacharya, and Dr. Sudha Bhattacharya and WWGM for giving me this opportunity, and Dr. Ashish Pal for the NMR data, Dr. Sam Mathi for the animal, for helping us in animal experiment, and Dr. Arkasubhra for providing us the GNE gene construct, and Dr. Sheetal Gandotra for helping us in cloning experiment, and the ICMR for funding, and the ACSIR for my registration, and the CSIR for my fellowship. Thank you.
Thank you so much, Ms. Rao. That was a very comprehensive presentation on the work that you are doing. Does anyone have questions? I don't see any in the chat box. Does anyone want to just raise their hand and ask a question?
Hi, Divya. Can you hear me?
Yeah.
Very nice talk. Nice talk. Just
Thank
I have one point for you. Just your nanoparticles are slightly positively charged.
Yeah.
And maybe you should look at the cytotoxicity profile of it once.
Okay.
Yeah. Okay.
Thank you. Any other questions? Okay, so maybe people can put questions in the chat box and Ms. Rao
Okay,
can...
I'll answer there.
Maybe I'll ask one.
Yeah.
This particles that you're making with the polymers that you have shown, and you want to modify that with mannitol.
Yes.
So, so that it gets taken up through endocytic pathway. Is that what you're saying?
So that's the one for, like, if we are able to check if it is going with the caveoli mediated pathway, then we are planning, if that's the case, then we'll be modifying it with the mannitol. But if it's not, then we have to come up with something else.
So the other question is that, you're seeing your gene actually a protein expression in the target muscles, right? But you think it is a little less. But endogenous level of muscle GNE is also quite less.
Yeah.
Do you have a ratio of what is endogenous and what have you given as exogenous?
No, I don't have that ratio. But so you are talking about the in vitro part.
Not only in vitro, the animal experiment in the end, last one
Okay.
slide that you showed where you said it's much less.
Yeah. So I am comparing it with the control where we have not given any nanoparticle or any exogenous.
No. See, I was more thinking whether you said that there's one animal experiment that you have done.
Yeah, it's not actually one. So we have tried with multiple
Okay, you
doses
have, and you are getting very low level expression. That's what you said. But what is the level of exogenous protein, the exogenous gene that you're giving expression from there versus what is endogenous expression?
Okay, that I need to check.
Yeah, please do that because generally GNE expression in muscles is much
it's
lower
low.
than other tissues. Okay.
Okay.
Because here, even if small expression can actually be useful,
Yes.
Okay, anyway, we can talk about it.
I just had a small comment, Divya. Excellent work. Maybe you could also use the luciferase construct which Rajesh is using for your initial standardization because it will give you cleaner results.
Okay.
The background due to GNE expression will not be a problem then.
Okay.
Okay. Thank you.
Thank you, ma'am.
So thank you so much, Ms. Rao, for for this work that's also again like so promising for patients, and we hope to have maybe something that's quite translatable from this coming soon. So now I would like to invite our next speaker, Dr. Arkasubhra Ghosh, who I don't think needs any introduction. I think most of us know him quite well. He's the chief scientist at the Grow Research Laboratory, Narayana Nethralaya Foundation, and he will be talking to us about his work on gene therapy. Over to you, Dr. Ghosh.
Thank you, Shilpi, and thank you, all of the and WWGM for having me today. Share this.
Thank you for being here and for all the wonderful work that you're doing.
Sure. My pleasure. Can you see my screen? Is it full screen?
Yes.
Okay. All right. So I'm going to talk a little bit about the AAV gene therapy work that we've done for GNE myopathy. It's all done under support from WWGM, so I'm very thankful for that. This is a photograph of the laboratory, which is now an approved laboratory to produce clinical grade AAV vectors. So adeno-associated viral vectors, or AAV for short, are extremely efficient vectors. And here in the video, what you'll see is these are epithelial cells, and they have been transduced with an AAV vector, and you can see the green protein coming up. So AAV vectors are known to have very high levels of expression, particularly for muscle delivery. AAV vectors have been the gold standard. And unless the doses are extremely high, they are not associated with very toxic side effects. Benefit of AAV vectors amongst other viral vectors is it's non-integrating, so therefore it is fairly safe compared to lentiviral vectors or gamma retroviral vectors. So in our lab, we work on different types of muscle diseases. the two primary being Duchenne muscular dystrophy and GNE myopathy, but we also work on other ocular diseases, blindness diseases, and sickle cell anemia and neurodegenerative diseases. So we primarily focus on transgene engineering, meaning in case of gene therapy, as all the other speakers have explained, you have a piece of DNA that the viral vector is going to carry into the disease site. So engineering them so that you have the best expression and regulating them so that you do not have off-target expression. These are our core fields of work. And then we are also now making clinical grade vectors for human trials and, of course, the associated regulatory processes. So this is broadly the muscle gene therapy studies that are ongoing in the lab, the DMD studies with both microdystrophin and minidystrophin. Those are ready for clinical trials. Hopefully, we will submit the clinical trial application soon, and I'll show you brief snapshots of that. GNE myopathy, of course, we did a proof of concept study a couple of years ago now. We are still awaiting some funding to take it forward. LGMD, the same status. We're waiting for funding to come through. And but in the meantime, we've developed some of these tools which can be applied to all these different gene therapies to make them more efficient, more safe. So I'll briefly touch upon that as well. So the first simple job was to construct a GNE expressing AAV vector. And we made that first, and we went on to check its expression in different cell lines. Here, I'm only going to show you the myoblast cell line C2C12. It's a mouse cell line. And you can see in this Western blot here that the GNE gene is expressed. And of course, in the cell line, you can't see this is a control, just to show that AAV vectors can be tested in these cell lines. We went on to make an AAV9 vector expressing the GNE gene, and that was injected in mouse muscles. And once the mouse muscles were harvested after about 8 weeks, we checked for expression of the GNE gene, which is expressed. We use the same antibody that is used in these Western blots, so I'm not too sure how well that antibody works on tissue sections, but nonetheless, it is able to detect AAV expression, and this is a Western blot from the same muscle tissues that's in the immunofluorescence to show a good amount of GNE transgene expression. We tested different antibody as well using an immunohistochemistry approach to ensure that the expression can be tested properly. So GNE does have some of these challenges of the reagents that are available. So this was done a couple of years ago. The things from a safety perspective is that we do not want off-target effects of GNE therapy, because the therapy is going to be a one-time therapy only, one time injected into your bloodstream, and it'll go to all the muscles. So therefore, we wanted to have a muscle-specific promoter, which is the SPC512 promoter. Again, this came from our DMD studies. And we went on to once again test whether it expresses in the mouse muscle cell lines, and indeed it did. We can see good amount of GNE expression from this construct as well. So that's where we are with the GNE gene therapy work at the moment. We have a muscle-specific promoter expressing GNE transgene that expresses well, but we need to have better transduction and immune evasion, etc., as well as large-scale vector production using bioreactors and downstream chromatography-based technologies in order to produce clinical grade vectors. So I'm going to touch on some of these points to tell you about the clinical readiness of our group. So if we are to go to a clinical trial, we have to prepare clinical grade batches and CMC documents for AAV-GNE constructs, which will then, once we get the CT11 approvals, we'll go into the GLP preclinical tests, following which it will go to a CT4 application. For the patient population, of course, in order to be eligible for an AAV gene therapy, neutralizing antibody testing and genetic validation has to be done. So genetic validation, obviously, most of the patients would have. The neutralizing antibody assays are also developed. I'll show you some information on that. And then we have to have these biochemical assays for assessing GNE function, measures for sialic acid, glycosylated proteins, MNAC, etc. And then, of course, work with clinicians to establish appropriate functional endpoints for such trials. So, first of all, improving expression. This comes from capsid design. We've already done the one for the genome. This slide, you can see these are muscles, mouse muscles, which have been transduced with the wild type AAV9 or 2 different mutants of AAV9 that we created in our lab. And you can see that there is significantly more expression. This is green fluorescent protein, a reporter transgene expressing it in the mouse muscle. And that is then validated by Western blotting as well. So you can clearly see that these capsids can deliver the cargo at very high levels. But what does it mean therapeutically? Now, I haven't tested this in GNE, but I'm going to show you 2 pieces of data from the DMD work, preclinical work that we have done. So this is an animal model of Duchenne muscular dystrophy, the D2-MDX model, and that does not express dystrophin. The red that you see on the slides, so this is dystrophin protein expressed in the sarcolemma of the mouse muscles. So you can see the control doesn't have much. So this is the AAV9 wild type, and you can see with the mutants, you have significantly higher expression throughout the muscle. But what does it mean from a functional point of view? So I will show you now an experiment where these are the MDX mice, which are being made to run on a treadmill. So the first lane is control. And this control has been injected only with saline. And then these 4 lanes have different doses and different types of AAV capsids expressing microdystrophin. So here you will notice that these 2 are low dose and these 2 are high dose. So you can see already the control is already tired. It cannot run anymore within the first 10 minutes. And you keep an eye on these. You can see that in the first lane and the third lane, the animals are again not able to run, but this is after 20 minutes. So these are the wild type capsids, which are lower expressing. So you can see that the mutant capsid expresses even at a low dose is able to generate more force. So this is complete. Well, I won't say complete. This is significant functional recovery of force in these animals that it is demonstrating. So using these sorts of chimeric myotropic capsids, we should be able to do a therapy at 10 times lower dosage than what has been done in the past in clinical trials elsewhere. Then we discussed about the antibody levels. So this is a study that was funded by ICMR. And we were looking at the anti-AAV antibody response that is present in the general population. So this is data from 410 individuals, which includes both healthy controls as well as those with genetic diseases. And you can see that the highest antibody response is with the prototype AAV2 capsids, and the lowest is with AAV9. So all the muscle data that you saw is with AAV9. That's what we had packaged the GNE also originally. And this is neutralizing antibody. That was total antibody response. This is neutralizing antibody response. And again, if you look at these bar graphs, so you can see that as the dilution of the serum from these subjects is done, the transduction levels don't improve significantly in case of AAV2, whereas you can see here that the transduction levels for most of the AAV9s are significantly higher. So that means that the AAV9 has less neutralizing antibody and significantly less total antibody response. So that's the right serotype to go ahead with for clinical trials. All right, so now, as has been mentioned by Rajesh and other folks, that AAV gene therapies are extremely expensive. They cost millions of dollars. So what are we going to do about that? We are going to make all the vectors ourselves. That's the solution that we came up with. So we have a clinical grade vector production facility. It's one of the first in the country to be approved for producing GMP grade AAV vectors. And within this lab, what we have built up is a quality by design approach to have a very good, consistent production of AAV vectors, which align with very specific critical quality attributes that help regulatory approvals by having consistency, quality, and safety inbuilt. So this is essentially our platform. We used a fixed bed bioreactor platform to produce vectors. And this is an example of the consistency of vector production processes across different serotypes within the lab. So there are each of these chromatograms that you see are 9 different independent runs done across 3 different serotypes, and you can see the consistency in production and elution. Of course, the yields are different based on the type of serotype that is being packaged. And also there are differences in yield between the intracellular versus the extracellular parts of it. So these are well defined now as processes. And safety and immune response. So these same vectors that you saw in the previous slide, these have been injected in animals, and we have extensively assessed the immune cell subsets, and I'm only showing you the CD8 T cell subsets. These are the cytotoxic T cells, often the reason for immune response. And if you just focus on the yellow bars, like this is the control, this is the saline injected one, and this is the AAV9 injected one, intravenous. The other ones are intrathecal and cerebroventricular. So you can see that none of them, with the AAV9, with the intravenous delivery, there is absolutely not much difference in response. The same goes for secreted cytokines, chemokines, and interferons. Again, I'm just showing you a small panel of this. We have tested close to 52 different factors. So with that, the final quality by design approach that we take, where we ensure that the vectors that we produce have the right titer, the integrity, they are non-aggregated. Here's an example showing aggregated versus non-aggregated. So the quality of the vectors that we produce is quite high. Then we test the potency, which is consistently performing. We have innovations around the formulation itself, which allows for stable, higher stability and higher transduction, purity, identity, and safety and sterility. So all of these we've developed in-house so that we can keep the overall cost of the vectors, vector manufacturing low. So the hurdles to cross in order to make gene therapy accessible is that the transgene expression levels have to be sufficient for clinical efficacy. We can improve capsid efficacy and production yields. Therefore, production costs can be affected. So the optimization areas are essentially vector design and vector production processes, and the solutions are optimal transgene design, high capacity, high efficiency capsid designs, and high yield process designs. So in a snapshot, that's what we've worked on and that's what we've built now. So we now have these indigenous technologies for AAV production with locally held IP and quality by design approaches that lead to increased expression of the vectors. So better delivery to the muscles. Therefore, you need lesser dosage in order to reach a clinically useful expression, clinically meaningful force generation. And overall, by doing QBD approaches and improving our AAV production processes, we have increased our vector production yields, and therefore we need to we can achieve the clinical scales without having to expand the production too much. So overall, all of these things together will reduce the cost significantly, and we hope that the cost will be somewhere around 1/30th to 1/100th of the international cost. So essentially, are we going to get there? Is the question that I get most often from patients. We have the capability. We still have to build capacity. And of course, can we reduce cost of gene therapies? Yes, potentially by 100 times. And can we do it tomorrow? No, because we need to have funding in order to establish a factory or industry scale vector production. But realistically, how quickly can a genetic disease gene therapy trial start? In our lab, given our current readiness, it can be done within 12 months, provided we have the support for it. So with that, I'd like to thank my lab, particularly the folks who have been working on gene therapy with me, and Shubhradev did the GNE myopathy work. And of course, all the various funding agencies, including World Without GNE Myopathy. Thank you so much. And thanks for your attention.
Thank you so much, Dr. Ghosh. And thank you for such an informative talk and also for answering a lot of the questions within your talk. And we hope that you will get the funding so that this work can really go ahead full steam. So we have time for one question? We're actually quite late. I just realized that. Although somehow everyone is speaking within their time, but somehow we're still running late. I'm not sure what's happening. So we have time for one question. Does anyone have a question? There's one question from a patient asking whether it will stop the disease or cure it, gene therapy, whether they can walk normally after gene therapy or not.
Short answer is I don't know. Only a clinical trial will tell that. But yes, gene therapies are curative. They are meant to be curative, whether you are doing AAV gene therapy or gene augmentation or using a gene editing approach, it's meant to be curative. But that having been said, depending on the stage of the disease that you are at and the amount of muscle damage that has already happened, the degree of curative function of the therapy is going to depend on that. So if you have lost significant amount of muscle already, or the tissue is completely fibrosed or something like that, that is not going to come back. So, but the myofibers that remain, the muscle that remains, that will regain strength. So some amount of curative effect will be there because your muscle will regain strength, so you will be able to do a lot more functions than what you were able to do without. So the earlier that you take the gene therapy, the better it is, and that is known from trials as well, that if you do the therapy early, you can have long-term curative effects. For the eye diseases, for example, it's now 16, 17 years, and there are folks who see just fine with that therapy. So,
hello, sir.
Yeah.
Sir, after gene therapy, we can walk normally or not, sir? It's after muscle damage.
Yeah, that's what I'm saying. So that depends on the degree of damage. So
sir,
if it is early on, when not enough damage has happened, then it has the potential to return you back to function. But once it's late, where there is significant loss of muscle mass, then it's difficult.
Okay, sir. Thank you.
Thank you so much, Dr. Ghosh. And also, I mean, congratulations on such an exceptional facility that you've set up. And
thank you, Shilpi.
And we hope that that you can continue this work for all of the rare diseases. So thank you so much. So now our next talk is also on a very interesting area. And we have Dr. Ravi Manjithaya, who's a professor at Jawaharlal and Nehru Center for Advanced Scientific Research. I believe that he is having, he has some issues, so he will not be able to give the talk at this moment, and that someone will be giving the talk for him. So I believe that's Ms. Ritu Provosen. Please, over to you, Ms. Sen and Professor Manjithaya. We're really looking forward to hearing about your work, especially since we are excited about possible applications in GNE myopathy as well.
Thank you so much, Shilpi. I'll share my screen now. Is my screen audible?
Yes.
Yeah. Okay. Thank you so much, Professor Sudha Bhattacharya and Professor Alok Bhattacharya, for giving us this opportunity to share our work. So today we will primarily be focusing on a particular project in the lab that we are currently looking into, which is a specific form of mitochondrial quality control pathway called mitophagy, and how mitophagy is perturbed in a spectrum of rare genetic disorders called mitochondrial diseases. And ultimately, we would like to briefly discuss the knowledge that we have gained from literature about this specific small molecule metabolite called urolithin that has shown extreme promising results in terms of its capacity to ameliorate several of the mitochondrial health damage that these diseases cause, and how we have now taken forward this particular small molecule in several of our therapeutic applications. So if you look at mitochondrial diseases, they are a category of rare genetic disorders caused either due to a mutation in genes encoded by the mitochondrial DNA or by mutations in genes encoded by the nucleus, which are critical for mitochondrial functioning. Now, these two spectrum of genes, when perturbed in either side, leads to diverse phenotypes in which causes mitochondrial diseases. And the phenotypes are multi-spectrum, ranging from neurological impairment to optic atrophy, ophthalmoplegia, severe cardiomyopathy, skeletal dysplasia, as well as defects in the neuroendocrine and gastrointestinal system as well. Now, while there is a organism-based damage to the entire organ system in such disease conditions, literature has suggested that in such disease scenarios at the subcellular level, there is accumulation of damaged mitochondria characterized by defective functioning in terms of its incapacity to perform electron transport and consequent oxidative phosphorylation, aberrant cristae structures, accumulation of mitochondrial DNA damage, accumulation of unfolded proteins, as well as a dysfunction in its metabolism. Now, the idea is that if there is damaged mitochondria accumulating, could it be because of severe disruptions in quality control of the mitochondria or not? Now, the cell senses damaged mitochondria, which is transformed from healthy mitochondria to damaged in the presence of excessive ROS, in the presence of mutations like nuclear and mitochondrial genome mutations, as well as drugs and genotoxic stress. Such damaged mitochondria at the subcellular level gives out several damage indexes, such as aberrant ROS, loss in membrane potential, defective OXPHOS, accumulation of mitochondrial DNA damage, and unfolded proteins. Such damage is very often recognized by several quality control pathways in the cell, which are as follows. For localized quality control, there are mitochondrial proteases at the inner membrane and at the matrix, which cater to unfolded and misfolded proteins. There is the ubiquitin-mediated proteasomal system for localized damage at the outer membrane. And ultimately, there's a mechanism by which there is pinching of small portions of damaged mitochondria, which are ultimately degraded in lysosomes. Now, while these processes are critical for localized quality control, in a condition where the mitochondria is damaged to an extent as is caused in mitochondrial disease-associated variants, we propose that there is a specific form of bulk quality control pathway of the mitochondria termed as mitophagy that sets in. Now, the common way by which mitophagy proceeds is by recognition of the damaged mitochondria, its engulfment in double membrane phagophores, which ultimately enlarges and fuses to form the mitophagosome, which transports to the lysosome, and in the presence of the acidic hydrolases of the lysosomes, the contents of the mitochondria are degraded. However, in case of mitochondrial disease-associated mutations, if there is a block at any step of this quality control pathway, then what will happen is that we will be reduced to a condition where the dynamic balance between healthy to damaged mitochondria shifts towards the damage, and this leads to an increase in damage accumulation in such conditions and further progression of the disease phenotype. Therefore, we wanted to ask if there are disease-associated mutations where there is a block in the mitophagy pathway at any particular step, could pharmacological intervention using modulators that could regulate the quality control of the mitochondria, principally by enhancing mitophagy, will that help in improving the phenotype that is associated with these disease conditions, both at the subcellular as well as the organ level? So to address this question, our project has a well-defined pipeline, which is now well established in the lab. We first identified mitochondrial disease-associated variants and tried to predict their extent of pathogenicity, following which we tried to assay for mitochondrial health parameters as well as the mitophagy flux in the cells in the background of these disease variants. Ultimately, if there's a mitophagy impairment, we try to delineate with exact precision where exactly in the mitophagy machinery such block lies, and ultimately use genetic or pharmacological modulators to rescue such block in mitophagy, and in turn check whether this helps in relieving some of the symptoms associated with such disease conditions. So for the first objective, the approach that we took forward is an holistic in silico screen from publicly available patient databases containing information about genes that cause mitochondrial diseases and their pathogenic disease-associated variants. We further use in silico approaches to predict, based on their structure, function, stability, and conservation, how much they deviate from that of the wild type. Once the pathogenicity is predicted based on these criteria, we generate these constructs and introduce them into an appropriate model system in case of cell lines for a preliminary study to understand the extent of damage it causes to dynamics and mitochondrial health, and consequently whether the mitophagy machinery in such disease conditions can effectively respond to such damage or not, which it usually senses under normal conditions. And we also are currently developing a C. elegans-based disease model containing the heterozygous knock-in of such disease mutations to study such damage as well as flux-based information in a model system. We are also, if there is a block, understanding using state-specific recruitment of specific mitophagy markers to assess where exactly the block lies and therefore to understand the mechanism, and ultimately use chemical biology and genetic approaches to check whether any defects in the mitophagy flux can be ameliorated. So for today's talk, we will primarily be focusing on the last stage of our pipeline, which is how to restore the mitophagy flux in the mitochondrial disease variants where there are severe impairments in mitophagy. So coming to this, I would like to mention here that since the 2000s, there has been a rampant rise in the number of mitophagy modulators that have been synthesized and available. Although there are several drugs that are present as enlisted over here, very few drugs follow or satisfy the criteria of a mitophagy inducer that can induce mitophagy without causing significant damage to the mitochondria. Now, if we look at the schematic over here, we observe that in case of dysfunctional mitochondria, which could possibly be accumulating due to the presence of mitochondrial diseases, would be engulfed due to the damage that it extrudes by double membrane mitophagosomes and ultimately degraded in the lysosomes, thereby maintaining cellular homeostasis. However, if such disease-associated damaged mitochondria keeps on accumulating in the cell without a clearance at a definite predestined duration, what would happen is that such mitophagy impairment would lead to their accumulation, and such accumulation would beyond a certain threshold lead to severe mitochondrial damage, causing mitochondrial outer membrane, inner membrane rupture, releasing several inflammatory and apoptotic molecules, and ultimately causing cell death. Thus, we could say that a mitophagy impairment in such scenarios will lead to further increasing the extent of damage phenotype associated with mitochondrial diseases. So therefore, from a literature mining, we wanted to actually find out one key molecule that could help us to induce mitophagy based on the following criteria. That is, it has evidences to show induction of mitophagy using the generic mitophagy flux assays that we use in the lab, has reproducible results in terms of mitophagy enhancement in cell lines and in vivo models, does not cause mitochondrial damage, and works specifically through the mitophagy pathway, and has also been tested successfully in disease mice models, as well as is currently now in clinical trials. So this molecule, urolithin A, is what we came forward with, which is a small molecule derivative of elagitannins and elagic acid, which is naturally produced by the gut microbiota. However, depending upon the individual microbiota composition, the bioavailability, as well as their conversion rate to urolithin A varies across individuals. And the initial papers about urolithin A was primarily done in C. elegans model system, where addition of urolithin A was associated with a consequent reduction in mitochondria-associated damage, which increased lifespan and also increased muscle strength and function in rodents. It has also been observed that urolithin A improves muscle function by inducing mitophagy in muscular dystrophy patients in terms of their muscle cells, as well as in muscular dystrophy mice models, by inducing key signature molecules of mitophagy and mitochondrial biogenesis associated genes. Mitophagy has also been shown to be enhanced upon urolithin A treatment in the presence of Alzheimer's disease, amyloid beta, as well as tau aggregates, and has improved cognitive deficits in several conditions. A recent clinical trial, which was a double-blind clinical trial, placebo-controlled clinical trial, has suggested that administration of urolithin A to individuals aged 40 to 65 years of age that showed low endurance in terms of their physical endurance capacity had an altered molecular signature of improved mitochondrial and cellular health, along with reduced serum levels of inflammatory proteins, as well as that of reduced levels of acyl carnitine, suggestive of increased metabolism as well as reduced inflammation. And currently, urolithin A has been commercialized into a product called MitoPure, which is the first clinically tested urolithin A supplement available readily as capsules that have been observed to show about 6 times more dose of urolithin A upon their ingestion in comparison to that of dietary supplementation. So based on these results, we wanted to check that in the disease variants that we are studying of mitochondrial diseases that lead to multispectrum phenotype, whether administration of urolithin A could actually cause any kind of improvement in the subcellular level on mitochondrial health parameters. So that's what our question was, that whether urolithin A could restore mitophagy in mitochondrial diseases. So this is a heat map representing each of the mitochondrial health parameters that we looked into in a combined way, which is we looked at the mitochondrial dynamics, we looked at the mitochondrial membrane potential, which is a critical signature of mitochondrial damage. We looked at ATP levels, ROS levels, and ultimately the mitophagy flux. And if you look at the wild type, when the wild type cells were treated with urolithin A, we found that under normal conditions itself, there seems to be a increase in the mitochondrial branch length, as well as an increase in the ATP levels way more than that of our controls, while the ROS levels were severely reduced and there was an increase in mitophagy flux. Now, compared to the wild type, when we administered urolithin A to some of the variants that we are studying, they showed promising results, and I'll draw your attention to one of the variants which showed that upon urolithin A treatment, there indeed, from a depolarized membrane potential, there was restoration of membrane potential. ATP level increased manifold, ROS levels reduced, and consequently, mitophagy flux seemed to be restored, which was severely impaired under non-treated conditions in this variant. So now that at the subcellular level, we are further probing into several other parameters to check what the effect of urolithin A is in terms of mitochondrial homeostasis. Parallelly, as I mentioned earlier, that we are also looking into several disease models with heterozygous knock-in of the disease mutation in C. elegans. And I'll share some of the unpublished data that we have observed. So we also added urolithin A in the food supplement of C. elegans disease models containing some of these variants. And we checked for several developmental indexes in these variants. And what we observed is that while there was no change in brooding capacity, it seems like the embryonic lethality as well as the larval lethality seems to be reduced in both these variants when they were treated with urolithin A. We are further characterizing this based on motility as well as replicating the defects found in cell line in these disease variants to further consolidate it. But to deviate from the results that we are doing, we currently found a urolithin A which was indigenously synthesized by Professor V. Ravichandran at NIPER, Kolkata, who had provided us this urolithin A. And when we compared the indigenously synthesized urolithin A from that of the commercial urolithin A that we were using for our experiments, we actually found a similar and perhaps even a higher level of mitophagy induction compared to that of the commercially available ones, suggesting that indigenously synthesized urolithin A also induces robust mitophagy, similar to that of commercially available ones, and therefore might be a more readily available source of urolithin A that we could use for our further research. So in summary, urolithin A has emerged as a small molecule which effectively causes upregulation of mitophagy and mitochondrial metabolism associated processes. We in our lab in the presence of variants have also observed similar results. And utilizing urolithin A in such disease-associated variants do reveal its critical role in disease phenotype associated with such diseases. However, the mechanistic basis of urolithin A's means of action, as well as whether it slows down disease progression in such scenarios, remains to be elucidated, and we are currently working on the same. And parallelly with urolithin A as our control model drug, we are trying to perform an FDA-approved library screen to find compounds that follow two criteria, that is, they do not cause damage to mitochondrial health, but consequently cause increased mitophagy, which could possibly bring in additional compounds that we could test in disease conditions of mitochondria where mitophagy is severely impaired. With regards to GNE mutants and GNE myopathy, we have observed that in literature that there are several GNE mutants associated with defects in autophagy, as well as defects in the activation of AMPK, which is the master regulator of autophagy. And it is interesting to find that urolithin A, although the exact mechanism is unknown, there are promising results suggesting that urolithin A activates the mitophagy axis through the AMPK pathway and can also activate bulk autophagy, and hence might prove to be one of the supplementary drugs that could be tested in such mutants. So thank you. I would like to thank our collaborators and as well as our funding agencies, and I'm happy to take questions.
Thank you so much, Professor Manjithaya and Ms. Sen, for delivering that talk so clearly, and it's exciting to see the kind of applications that this has. And also, I mean, yeah, the small molecule therapy is something that can actually come into the market really fast. So we're really keeping our fingers crossed that this work can have like tremendous applications for different rare diseases. So we have time for a couple of questions. We are running a bit late, but happy to have a couple of questions. Would anyone like to raise their hand or put a question in the chat box? I don't know if I don't know if Professor Manjithaya can actually like answer the question, or maybe he can just put it in the chat box. Okay, so maybe we can have questions put in the chat box so that Professor Manjithaya can then answer it in the chat box itself. That might be better. It does sound like you're quite ill. I'm so sorry.
Yeah, very sorry that Ravi is not able to speak, but your student has done a very excellent job. So we really understood everything, and urolithin, because it's not toxic, is already being used as a supplement. So we hope it can be really very rapidly come for clinical trial. Yeah, yeah. So we hope that it'll very rapidly we can move to the stage of clinical trial. But before that, we should do some testing in GNE model. Yeah.
Yeah. Actually, this is some of the questions that many patients have been raising in the past about mRNA therapy as well as gene therapy, whether it will cure. And generally, the damaged muscles may not be able to regenerate. But is it possible that urolithin A combination therapy, if it works along with another treatment that actually targets the disease directly, that could be a potential thing? And this is something that we are following up with ICMR. So that's the whole idea about thing. If it shows some promise in GNE models, I have a feeling that it will show. And since it is absolutely non-toxic, though I think a clinical trial can definitely be done. And I think we most likely that ICMR may give us money to do this trial, along with 6-silyl lactose as a possible direct therapeutic molecule. It's a great job. And as Sudha said, you did a good presentation.
Hey, I have a question. Hi, Ravi. Hi. How are you? Okay, fine. Okay, so great talk. I just wanted to ask you one thing. What is the mechanism behind urolithin? How it is modulating mitophagy?
It is not very well known exactly what it does, but the parameters by which mitochondrial biogenesis happens and also the entire genetic machinery of mitophagy, what has been known, is really encapsulated by treatment by urolithin.
Okay.
There are 1 or 2 papers only that talk about some targets, but people in the field believe that there is no target yet identified yet.
Okay.
So, and it's like a polyphenol in some sense, so it may not work through a single target as well.
Okay.
But this is what they feel. And there is a paper where genetically, if you induce mitochondrial biogenesis or mitophagy, so you have a, let's say, an expression profile of all the genes that turn on during mitochondrial biogenesis and mitophagy. You can compare that with cells that have been treated with urolithin, and there's a considerable overlap.
Okay. Somehow it is bypassing and actually enhancing biogenesis as well as phagy, right? Some kind of proteins.
Absolutely.
Must be some kind of a, if like either it is acting directly on protein or maybe through the transcription machinery and upregulating the proteins.
I think there's no one mechanism of action. In fact, even ATP production and decrease in ROS levels and subsequently mitophagy, all of that is induced. It's just too good to be true. And that's why people are getting excited. That company in California that is selling that product now is telling people that they will live longer, even as a longevity molecule. They're selling it, and
yeah,
if you subscribe to them, they will send you urolithin to your home, at least in the US.
Okay, great.
Many of the muscle degenerating diseases, though mutation could be wherever it can be, finally, I think somewhere or other mitochondria gets involved, right?
Yes. So to answer your question, what you were asking previously, what we think is, irrespective of whatever gene therapy will come for any mitochondrial disease associated gene, there will be a supplementation like urolithin to go along with it. And I'm pretty sure that will be beneficial. You will have some amount of synergy there.
Okay.
Thank you.
Thank you so much. We are running a little bit late. We were supposed to take a break, so maybe we can take a 10-minute break and we can come back at 6:45 for the international uh, of our event. Thank you so much, Professor Manjithaya and Ms. Sen, for that excellent talk. So we'll take a break and we'll be back at 6:45. Thank you very much. Hello, Michaela, are you there?